that this is a very, very complex research field. There's sort of a lot of controversies, and nothing is really as straightforward as sometimes we would like it to be, or maybe people studying these things would like to make it out to be. And that kind of brings us to the last little topic that I'd like to talk about today. So you clearly, are skeptical of, a lot of research going on in this area, rightfully so. And you've been very outspoken over I would say maybe the past few weeks on Twitter, posting kind of multiple Twitter threads in sort of this development, this debate, maybe we'll call it, regarding, and, we'll just put the name out there, but, a well-known scientist, Dr. David Sinclair, who's kind of very, very outspoken in the longevity field, has gained a lot of popularity, and done a lot of, research. He's a very, well-trained scientist. So can you just give us a brief intro kind of into what you've been so outspoken about on this topic? It relates to, the quote longevity genes that we briefly brought up. It does include a little bit of NAD, which we've extensively talked about. But can you just kind of give us a brief overview of what some of your issues are with maybe not only Dr. Sinclair's work, but maybe some of the claims that he is making about his work, and then we can kind of build upon it from there.
Well, people are interested in longevity. People are interested in lifespan. In fact, Lifespan is the name of a best-selling book by Dr. David Sinclair that has been incredibly influential. I would say that David Sinclair is the most influential person in the molecular biology of aging.
Yeah, I would say that's accurate,
right?
For sure.
And globally, and he's been a scientific leader, and he's now basically introduced people around the world to the ideas of molecular aging. So if you look at the front cover of the book, the subtitle is "Why We Age and Why We Don't Have to," and unfortunately, Dr. Sinclair doesn't know why we age, and has suggested that we don't have to, in a way that is actually counterfactual. And the whole premise of the book is counterfactual. If you look at the back cover of the book, it says, what if aging is a disease, which is a counterfactual question, and that disease is treatable, which is also a counterfactual question. So the whole thing is based upon a premise of, what if you didn't have to age? What if you could live forever? Wouldn't you want to? So he's using basically a literary technique that was described by Samuel Taylor Coleridge of temporary suspension of disbelief, that he's taking people on a journey in an imaginary world in which we understood aging, and we had monogenic longevity genes that we can activate such that we don't have to age, and aging is now optional because we understand aging so well. Well, if that were the world, then you might consider that aging is treatable. And if aging is treatable, then aging must have been a disease. And so he's got people all, mixed up into thinking that he understands why we age and why we don't have to. The problem is the underlying evidence in the book is all based upon stuff that doesn't work. So, the fundamental insight made by Dr. Sinclair in 1997 was how a yeast mother cell can divide so many times in one model of yeast aging called replicative lifespan. And this is a model in which calorie restriction, growing the cells on lower glucose, gives that mother the ability to divide more times, to produce daughter cells more times. The other model of yeast aging, loss of SIR2 gives a longer lifespan. As I've explained on Twitter, in the model of aging that Dr. Sinclair and Guarente were working on in 1997, the advantage is only to the old mother cell, a cell so rare, there's only 1/2 to the 21st power of those cells in the culture. In other words, there's no survival advantage of the yeast culture to even maintain the function of that SIR2 gene. And loss of that SIR2 gene gives cells that have a longer lifespan in the other model, literally the model in which the cells stay alive for weeks or months. Loss of SIR2 has an advantage. SIR2 is not even a longevity gene in both models of yeast aging. And then David, unfortunately, he and I are on a first name basis. We know each other very well. I'm actually in his book for my discovery of the nicotinamide riboside kinase pathway. At one time, I consulted for his company, so I know a lot about the resveratrol story as well. You know, David is always saying that SIR2 was then found to be a longevity gene in worms. It was found to be a longevity gene in flies, and then it's a longevity gene in mice and in people. And the problem is it was reported to be a longevity gene in worms and flies. And then 10 years, it took 10 years for that literature to be partially cleaned up by Linda Partridge and investigators from 9 institutions that showed no, it was a strain effect. It's only in particular backgrounds of worms and flies that extra copies of SIR2 are lifespan extending. When you outbreed and you do that in, you know, fresh worms and flies, there's no survival advantage. As I said before, there's no reason that you would expect a gene that is important in 1/2 to the 21st power of yeast cells to anticipate the role of thousands of genes in animal longevity, when longevity is an emergent property of animals and is not even a directly selected thing. So the whole idea doesn't make sense from the evolutionary biology of aging. It's inconsistent with the recessive nature of growth factor longevity genes, which are not pro-fitness genes. The resveratrol is not a SIRT1 activator. The things that he still claims are SIRT1 activators, he actually still claims that resveratrol is a SIRT1 activator. Those don't help people. They were extensively tested by GSK after GSK bought out Sirtris and all of the technology that was developed by Sinclair. And he basically acts like, you know, the longevity genes were discovered, they were validated in animals, they exist in humans, they can be activated. He claims to have, you know, rejuvenated vision in mice. As I said, the experiment wasn't blinded, it was pre-announced, which makes it, you know, not really believable. And the problem is that rather than, when I bring up these critiques, which are shared broadly in the scientific community, you know, Dr. Sinclair has not really responded. And most recently, he claimed that misinformation about resveratrol was being propagated by myself. And he responded by saying, oh, this will clear it up with a 2006 paper, which is, you know, 16 years behind the times of refutation from other people. So at this point, you know, he's blocked, you know, Dr. Jeffrey Flyer, who's the former dean of Harvard Medical School. He's blocked me. He's blocked Dr. Brad Stanfield, who is a, you know, person that, you know, has a YouTube channel who talks about longevity and the evidentiary basis for scientific claims. And you know, he has an audience from the book and from Instagram and from a podcast that I'm concerned is spreading a lot of misinformation. And so I think it's a real problem.
Yeah, and I mean, there were, I've been aware of kind of all of the recent blocking and things like that. And I remember seeing a tweet from even the former dean of Harvard Medical School who had quoted Dr. David Sinclair's tweet about, oh, hopefully this will clear it up, where he cited his 2006 paper. And the dean even said, it's very troublesome that you're citing a paper from 2006 that hasn't been replicated or has even been maybe disproven by some more literature in recent years. I mean, that's a paper from 14, 15, 16 years ago. And so, yeah, it seems like it's, a sort of attempt at salvaging, this whole sirtuin as a longevity gene story. And the resveratrol thing as well, it's, as you said before, this whole lifespan book and kind of a lot of the claims he's making is all based on this foundation that we have to accept sirtuin as a longevity gene, and that if we can activate these sirtuins through whatever mechanism, whether it's caloric restriction, fasting, resveratrol, that we can extend lifespan. And sort of if that's not true, then this whole sort of, it's all a kind of facade, and this whole sort of foundation breaks down. I mean, is that correct?
It's the book is falsely premised is the problem. And like I say, I don't think that aging is particularly well understood. I think it is a part of life. I think the dominantly acting longevity genes is a myth. The sirtuin activation story is not true. And that even and then importantly, the idea that sirtuins are the primary mediators of NAD is also not helpful. There's hundreds of NAD dependent processes. There's even dozens of NAD consuming genes, and among the NAD consuming genes like the PARP1, PARP2, and the 5 PARPs that we found that mediate the interferon response in coronavirus infection. There's enzymes called SARM1 and CD38. All of those gene products are more highly regulated than sirtuins. Excuse me. So I don't think that, I think that sirtuins are, moderately interesting. There's been tens of billions of dollars invested in looking at them. We know a bunch of things about metabolic regulation that because of that work on sirtuins, but there's way too much. The street lamp has been lit very, very brightly around CERT 1 through 7. Initially,